So welcome everybody to this podcast about computational modeling of cardiac cellular electrophysiology. My name is Niels Vogt from the Institute of Pharmacology and Toxicology in Göttingen, Germany. I have with me today Dr. Jordi Heijman from the Department of Cardiology, Maastricht University in Netherlands. He is a distinguished expert in cardiac cellular electrophysiology and computational modeling, and he uses his models to integrate knowledge from ion channels, molecular pathways, to understand the interactions and to determine the contribution to cellular arrhythmias. So he will give us an introduction about computational modeling. So Jordi, thank you for coming here.
Thank you for having me.
When did you first get fascinated about computational modeling, and what is still fascinating you about this technique?
So I have a background in applied mathematics, and the question is, where do you apply it? And as soon as I came into touch with biological subjects, I always felt that this is a field where an insight in computer modeling and mathematics can really help us to better understand the biological problems and can really add something to our understanding. And so that's when I really got fascinated of using this knowledge in applied mathematics and applying it to the field of biology. And in particular, the opportunity came to do this in the field of cardiology. And I could really see how the models can help us to better understand the mechanisms of arrhythmias in patients. So ever since then, I've been very fascinated about applying this tool in this field, and it still fascinates me today.
So you mentioned that you are a mathematician from training. Is being a mathematician or a computer scientist a requirement to do computational modeling, to learn computational modeling?
No, I don't think you necessarily have to be a mathematician to do this well. We have examples of people with a background in pharmacy or other biological subjects who've also learned this. And in general, I see ourselves as translators. So we try and take the biology and translate this into mathematical formulas and computer models. And of course, a basic background of mathematics helps with that and is also necessary. But in general, if someone is motivated enough, they are able to pick up these tools along the way. And I think indeed that there's also various tools available now that help in this process so that you don't always need the full mathematical detail of implementing a model to still get useful answers from your simulations. So overall, a bit of mathematics is required, but you don't have to be a hardcore mathematician to do this.
So if you decide to do computational modeling, can every student do it on his office PC, or do you need special hardware to do this?
Of course, it depends on what you want to simulate. We have models that range from subcellular events to whole patient simulations. And of course, the more complex the model becomes, the more expensive the computational requirements are. But we see that a lot of the simulations that we're doing at the single cell level, the computer hardware has gotten so good that even a regular laptop or a desktop computer is able to do this. Of course, if we go to the more complex multicellular simulations, there are some tools using graphical processing units that allow to do this. But if we make it even more complex, then we really need to go to the specialized hardware. But for first simulations, to start with, a regular computer will do just fine.
So if an early PhD student would like to learn computational modeling, what may be a good starting point to do this? What software should they use?
So I think, first you need to understand the cardiac electrophysiology a bit, because that's in the end what you will implement into the model. And then when it comes to the actual techniques for the modeling itself, there's certain tools that are available that allow you to really make some first steps fairly easily. Actually, in my presentation and in the demonstration we will do afterwards, I will highlight a number of these, including the MyoKit software package, and that is one example that people could use to get a first feeling of it. There are certain examples available of models that they can download from that website and that they can fairly easily, for instance, simulate the effects of a reduced expression of a channel. And that's something we will show afterwards as well. So I think that would be one example of a tool that people can use to get started with this.
So finally, going from the status of a PhD student to a bit more advanced state and developing computational models in the future, do you think that computational modeling, advanced computational modeling, is in the future able to replace actual experiments in animals, in myocytes, in patients?
I think computational modeling is very much complementary. So I don't think it will necessarily replace experiments, but one thing it will probably do is help focus experiments. So by running simulations beforehand, we know which parameters to assess or strongly to control in experiments, and it will therefore make our experiments more focused. Of course, we also always need to validate our models, and experimental data are very much necessary for that. So that's another reason why they won't completely replace it, but will be in addition to it. But on the other hand, models might allow us to do certain things that we cannot yet do experimentally. Either because we don't have the right drugs or the genetic models are not available. Computational models might help us to go one step beyond the experiments. So complementary, they won't replace them, but together I think they can really help us to improve our understanding of cardiac electrophysiology.
Thank you very much, Jordi, for this interview. And we are going to listen to the presentation from Dr. Heijman. And after the presentation, he will give us a demonstration of how you can use computational modeling and how you can start computational modeling.
So I would first like to give you a bit of motivation why that would be useful. And what I've depicted here is just a PubMed query showing publications first in cardiac electrophysiology. If we go to the next step, it's the computational modeling of cardiac electrophysiology. Now keep in mind, these are two different scales. There's about a factor 10 between them, but you can see that the increase in publications dealing with the modeling of cardiac electrophysiology is rapidly increasing. And if we calculate the ratio between both of them, you can see that about 10% of publications in cardiac electrophysiology now include a computational modeling component. So there is a decent likelihood in the course of your career you will be involved in projects where computational modeling plays a role. And hopefully, based on this talk, you will at least get some of the vocabulary and know some of the concepts that will facilitate the discussion with people who do computational modeling. And maybe for a few of you, it will also inspire you that actually it's a very interesting way to look at cardiac electrophysiology, and that it might be something you want to do yourself. So what I want to discuss in this presentation are four main topics. I want to give a little bit of the biological background and the motivation for computational modeling. I want to describe the most commonly used methodology, provide some overview of historical landmarks in this field, and then give you an idea of some of the tools that are available that actually allow you yourself to start and play around with this. And finally, we will come to some of the current limitations and of course also the future challenges that we currently have in the field of computational modeling. So to get started, a little bit of a background on cardiac electrophysiology. Probably well known for most of you, but to get everyone on the same page, if we talk about cardiac electrophysiology, particular cellular electrophysiology, the main mechanisms driving the electrical activity of the heart, the central element in that are ion channels. These are intricate multi-protein complexes. We modelers like to depict them like this. They are much more complicated than that. They have pore-forming subunits, they have regulatory subunits, and they're a very dynamic environment that control the electrical properties of the cardiomyocytes. In their open or conducting state, these ion channels allow very specific ions to either enter or exit the cardiomyocyte. For instance, here in blue, you can see a sodium channel. And when this channel opens, sodium ions will go from the extracellular domain into the cell. Well, this is a movement of charge. That means a current, and because we get more positive charge inside the cardiomyocyte, this leads to a depolarization. On the other hand, we also have potassium channels. If these potassium channels open, potassium ions flow out of the cell, and we get a repolarizing current. So these currents shape the membrane potential. However, what is very important is that all of these ion channels, or most of the ion channels that are important in the heart, are also voltage dependent. This means there's a feedback mechanism. The ion channels change the membrane potential, and the changes in membrane potential then affect how the ion channels behave. So most of the ion channels, for instance, open in response to a depolarizing membrane potential, and this is generally because they have charged amino acid in their transmembrane domains that sense these changes in membrane potential. But each of these channels does this slightly differently, does it at different membrane potentials, does it with different speeds, and has other different properties. And so millions of years of evolution has shaped a very interesting complement of ion channels, each with exact properties to really get the best shape of the action potential for the different regions of the heart. So So which are the ion channels that we need to think about in this case, a ventricular cardiomyocyte? Well, at resting conditions, the cardiomyocyte has a resting membrane potential of around minus 85 millivolts. And when this cell gets activated, either because we have a pipette sticking into it and produce a depolarization, or because it gets activated from a neighboring cardiomyocyte, this activates the sodium channels. I already told you on the previous slide, we have much more sodium outside of the cell than inside of the cell, so this produces an influx of sodium and a rapid depolarization of the membrane potential. I also told you that all ion channels are voltage sensitive. So this change from minus 85 millivolts to about 20 millivolts is sensed by all the other ion channels and causes some of them to open. For instance, it will lead to activation of so-called L-type calcium channels, and calcium will also flow into the cardiomyocyte. Now, this calcium plays a critical role in a process called calcium-induced calcium release, triggering a much larger release of calcium from the sarcoplasmic reticulum through ryanodin receptors, and it is this total amount of calcium that produces the systolic calcium transient that will in the end bind to the myofilaments and produce the contraction of the cardiomyocyte. So there we have two important ion species, sodium channels, calcium. Potassium channels are activated in parallel. We have a transient outward potassium current. But in contrast to sodium and calcium, we have more potassium inside of the cell than outside of the cell. So when these channels open, potassium will leave the cell and will produce a repolarization. The membrane potential will become more negative. This first happens with ITO, that's the first one to activate. Later, the delayed rectifier potassium currents activate, and this produces a specific shape of the action potential. We get a certain balance. We have a number of channels that try to repolarize the cell. We have some channels that try to depolarize. They form an equilibrium for a while. But as I said, each of them has their own kinetics. Some take a bit longer to activate, some a bit quicker. So in the end, the potassium channel takes over, and we get a full repolarization of the action potential. So electrically, the cell is back to its resting condition and would in principle be ready for the next action potential. But of course, a lot of ions have been moved around to get here. And in order for the heart to do this millions and millions of times, we also need to re-equilibrate these ions. So we need to have processes that take up all the calcium that was released through the ryanodin receptor. That's where the SERCA comes in. We need to extrude the calcium that came in through the L-type calcium channel. That is done by NCX and the plasma-lemmal calcium ATPase. We also need to restore a balance of sodium and potassium concentrations. That's where the sodium-potassium ATPase comes in. So you can see this starts to become a fairly complicated picture with a lot of ion channels that all influence the homeostasis of ions and membrane potential. Fortunately, it gets a lot more complicated. All of these ion channels are highly regulated. They're all phosphorylated on multiple positions. They can be oxidated. There's various forms of post-translational modifications that can occur and various pathways that regulate this. Nonetheless, this has given it its stability. That's why it works very well. But this also makes it difficult for us to intuitively understand what happens now if some of these components are changed, for instance, under disease conditions. So the complexity of the heart makes it difficult to intuitively integrate all these individual components and really understand, for instance, what are the mechanisms that produce an arrhythmia. Well, we can do experiments to try and elucidate this, but it's experimentally challenging to very selectively probe these individual components. We have not really the right tools always, pharmacologically for instance, to very specifically isolate each of these individual currents. For some we can do it, but not for all of them. Okay, we can try it in genetic models. We can knock out this channel in a mouse. But if we do this in a chronic situation, we often observe compensatory effects that might not be directly related to the change in this one specific ion channel. And even if we can selectively modulate one specific ion channel that we're interested in, we can only generally observe one thing at a time. We can either set the membrane potential and record a current, or we can record action potentials. But it's difficult to do both at the same time. And that's where computational modeling comes in. Computational modeling is a complementary approach that can help us to overcome some of these limitations. It might allow us to extrapolate and integrate experimental findings. For instance, we can know that an effect of a mutation has a certain effect on an ion channel, but what does it do to the action potential? Modeling can help us find that out. It can help us to determine the relative contribution of different pathological processes to a given phenotype. If you think about heart failure, atrial fibrillation, other complex diseases, a lot of these ion currents that I just showed you are changed. But which are the ones that really cause a problem? Well, with computational modeling, we can get insight into the ones that are predominantly responsible for the phenotype that we see. And I believe that in the future, modeling can help us to guide either personalized therapy or, for instance, test the safety of new drugs under a wide range of conditions. And that this future might not be very far away. I want to introduce to you two examples for that. The first one is the so-called CIPA initiative, so the Comprehensive In Vitro Proarrhythmia Assay. And this is an initiative that has been proposed by the Food and Drug Administration, the FDA, as a better way to stratify the risk for new pharmacological compounds that are being developed. Of course, if we develop a new drug, we want to make sure it's safe and that it doesn't have any proarrhythmic side effects. There's various tests that a compound needs to undergo to ensure its safety. With this CIPA assay, computational modeling now gets a very important role in this process, because this initiative says, well, we should first measure the effect on individual ion currents, and then use a computer model to integrate all of this information to see what happens to the action potential. And only thereafter, we will validate this, for instance, in human stem cell-derived cardiomyocytes, or look at a more clinical phenotype. So computer modeling is now officially recognized as having an important role for this drug safety testing. The second example has been highlighted last week in the European Heart Journal, in the Cardiopulse, a three-dimensional virtual heart tool. This is a tool that has been developed at Johns Hopkins University in the lab of Professor Natalia Trajanova, and she could show that these whole heart virtual hearts were very helpful to predict the risk of patients for developing arrhythmias. And in fact, these models were better than the standard risk assessment options that we have available. So these are two extremes of the scale. One, cellular work, drugs, patient risk in an intact patient on the other end of the scale, but both highlight a critical role for computational modeling to really better understand how the heart works in health and disease. So hopefully that serves as a good motivation that it's actually useful to know a little bit about computational modeling. So then a little bit of background. Now if we agree that it's important, what can we learn from it and how can we use it? Well, if I would write down what a computational model of cardiac electrophysiology is, then I would say that it's usually a set of ordinary differential equations which describe the dynamic evolution of various states controlled by a number of parameters. But that sounds rather abstract and might not tell you that much. So as an analogy, take the bottle of water that you see here on the screen. And if I take a bottle of water and I turn it upside down, then of course the water will flow out of it. But I can actually calculate what happens to the amount of water in this bottle. If I know how much water is in there, if I know certain parameters, for instance the size of the opening, then I can calculate how much water leaves the bottle at any given moment, and therefore I can update how much water is actually in the bottle. And I can do this over and over again until the bottle is empty. Well, the same holds true for a cardiomyocyte. If I know what the current membrane potential is, I can use that to calculate the ion currents. And as I told you, the ion currents produce a change in the membrane potential. So I can use that to update this. And then with the new membrane potential, I can calculate which effect it has on the ion currents and use that to update the membrane potential again, and so on and so forth. So if I want to develop a model, I need to do three things. I need to determine an appropriate model structure. Which are the ion currents that I'm interested in? Which should be integrated? What is the structure of the cardiomyocyte that I'm looking into? And these things will in the end determine which are the things that I will follow over time. Then the second step is to identify the parameters. For instance, the opening of the bottle in the previous example, but in principle it tells me how does the membrane potential produce these currents. And once I have these two components, then I can use this iterative manner to simulate action potentials, calcium transients, signaling cascades, or any of the other properties that you might be interested in
in a cardiomyocyte. So that's the overall structure that we use to simulate electrical activity and electrophysiology of cardiac myocytes. Now let us zoom in a little bit. Let us look at the individual ion currents. In general, we believe that an ion current is determined by the conductance, the open probability, and the driving force. What does this mean? Well, the conductance is simply how many channels do I have. If I have more channels, I will get a larger current. And it's influenced by the single channel conductance. So how easily do ions flow through the channel when it's open? These things in our models we generally assume to be fixed, so we don't really need to worry about them once we've set a certain value for them. The driving force is determined by the difference between the membrane potential and the Nernst potential, and is also a standard physical property. So again, there we don't have a lot to choose. So what modeling really is about is to think about how is the open probability of a channel changed. And we have three main approaches that we use to simulate that. And I want to highlight all of these three briefly to you. The first approach is what we call an instantaneous model for channels that change so quickly that we're not really interested in their dynamics. In this case, the open probability is just a direct function of some parameters. Best example I can give you is the potassium channel IK1. And basically, because this channel activates very quickly, we simply say, okay, we currently have a certain membrane potential, and based on the IV curve that was measured experimentally, we can say that for, in this case, minus 80 millivolts, we get a certain IK1 current. So we have an input VA1, we just have simply this relationship, and that produces us IK1. That's all. Then we're done with that current. Unfortunately, we cannot use that for all currents, because as I told you, each of them has their different kinetics. Some take a very long time to open, so we need to incorporate these dynamics. And Hodgkin and Huxley, which I will talk about in a few slides, developed a method to do this. And what they said is, well, there's different processes that control an ion channel. And again, here it looks very complicated, but if you want an analogy, think about doors in an airport. You can see two doors here, and only when both doors are open, people can go through. Same thing holds true for an ion channel. There might be an activation process and an inactivation process, and only if both are set in the right way, ions can flow through this ion channel. And also, these gates can respond to different things. I mean, if you think about a security door in an airport, some might open just regularly when you show up. Others, you might need to swipe a card or have a fingerprint reader. Same thing holds true for the ion channels, that different gates can respond to different processes. But, and that was the limitation of the Hodgkin-Huxley approach, they're all independent. So one door does not know what the other door is doing. Each of them controls the flow, but they do it independently. Well, later on, we realized that there's actually processes where that doesn't hold, that we do need this interdependence between both. That brings me to the last group of models, the so-called Markov models, and there this interaction is explicitly taken into account. So for instance, we know that a channel can only be blocked in the open state. So only when we know something about the first gate, something applies for the second gate. But with these three modalities, these are used in most of the cardiomyocyte model, we can capture most of the behavior of the individual ion channels that we have identified to be present in cardiomyocytes to date. So that is a bit of the general structure and how we modulate individual ion currents. Now, if we take these two together, then we go back to the level of the cell, and we go back to this action potential, which I introduced to you a few slides ago. And I already mentioned Hodgkin and Huxley. After them, we named the second approach to modeling ion channels. And they had also developed a mathematical model to explain the behavior, or the electrical behavior of nerve cells in a squid giant axon. And they already did this in 1952. And based on their quantitative work, really representing an excitable cell as an electrical circuit, they were able to obtain a lot of understanding in how these cells actually work and how the electrical behavior of this cell is regulated. And for this work, they received the 1963 Nobel Prize in Physiology or Medicine. What I wanted to do, I don't want to go into all the details, but I wanted to show you this is their entire model. This is how they describe the electrical behavior of the nerve cells. And there's a few currents here. You can see a sodium current again, some potassium current, some other ones. But you can see that it's a fairly limited set of equations. The mathematics are not that complicated of this model. Nonetheless, they were able to very nicely reproduce the action potential in these nerves. We have a measured one, and we have a simulated action potential. And at various conditions, they measured, did various measurements, which are the symbols shown here. And the model was able to reproduce all of these measurements, as you can see from the lines. So this was really the foundation for simulating action potentials. Fairly simple model, but very powerful in reproducing some of the key electrical properties, in this case, of these nerve cells. But of course, neuronal action potentials are quite different from cardiac ones. Cardiac action potentials are much, much longer. And originally, the ionic basic for this difference was incompletely understood. So modeling work helped us to realize some of these differences. And experiments in the early 1960s showed some of these things that could play a role. And Dennis Noble was one of the first to construct a model that specifically simulates the action potential in the cardiomyocytes, or in this case, in multicellular preparations. And he showed indeed that some particular changes that were identified could indeed explain the differences in the action potential that were also measured. So this is where the modeling already started to help us understand and make things plausible. We've measured A and we have measured B, but does A really lead to B? Models can help you to make that argument more plausible. Well then, of course, since the 1960s, there have been extensive experimental innovation. We now know how to measure calcium transients. We can isolate cardiomyocytes. We have a variety of techniques to really look into detail. And of course, all of this data that has become available has provided the basis for what one could call an explosion in computational cardiomyocyte models since the 1990s. And as one example of this, that sort of represents the standard model that already integrates many of the components. It was published in 1994. I want to show you the Lua-Rudy model. This is a model for the guinea pig ventricular cardiomyocyte, and is considered one of the first modern cardiomyocyte models. So instead of just the 2 or 3 channels that I showed you previously in the Hodgkin-Huxley model, we now have a whole host of them, and most of them I introduced to you when describing the action potential. We now also take into account that these calcium concentrations change and that other ion concentrations change over time, and that we have these transport mechanisms to achieve that. And so this model is still commonly used to date and really has served as the basis for then developing much more specialized models for different species, for different regions of the heart, and so on and so forth. So this is really the first modern model that serves as a basis for a lot of the work that we still do to date. So now you have a little bit of a background of what models there are and sort of which components are involved. Now, how can we work with them? What are some of the tools that are available to analyze cardiomyocyte models? Of course, in addition to developing these new models or developing a model for your specific disease condition, several people have also looked at specifically developing these more general purpose tools. For instance, to allow us to compare different models, thereby selecting the best one for the questions that we're interested in. And also allow for a more straightforward application of computational models by people who not necessarily have a strong background in computational modeling. I want to highlight 3 relevant recent examples of such tools. One is the Cardiac Electrophysiology Web Lab, the second is MyoKit, and the third is ECG Sim. They all operate on various different ends of the scale, and just like with computational models, there is not one size fits all, but there's a variety of tools available that, depending on the questions that you want answered, might help you in your research. First, the Cardiac Electrophysiology Web Lab was really meant as a tool to compare different computational models by separating out what represents the model from what can I do with the model. I can look at different experimental protocols. So previously, each model was published and it was validated or used for a number of different protocols. And they said, well, maybe we can split this. We have a number of models and we have protocols, and then we can arbitrarily apply a model to a certain protocol, and we can see whether that fits the experimental data that we have. So this basically functions as a big database where users can submit models and protocols, results are then calculated, stored, and can then be visualized. But it's not dynamic. They're just calculated, the results are fixed there, they're in this database, and you can visualize them. So it's not a computational tool at present per se, nor directly usable for model development or validation, but a very good tool to quickly visualize differences between available models. The other end of the spectrum, we have ECG Sim. This is a simulation program that enables one to study the relationship between electrical activity of cardiomyocytes and the resulting EKG. And here's just a screenshot shown here. And what you can see here is the action potential, and these sliders allow you to change various properties of this action potential in different regions of the heart. And you can see the results on the standard 12-lead EKG immediately here on the right-hand side of the screen. So again, you don't necessarily incorporate these individual ion channels. There's not an explicit model of that. But it's a very useful simulation tool if you want to know, well, certain things change to my action potential. I don't know how it happens, but I know that this is different. Does that match the EKG that I see in the patient that have this mutation, for instance? That's where this tool would be useful. And thirdly, and in between there, we have MyoKit, which is a real software tool for modeling and simulation. So it's much more like a programming language than the other two tools that I've shown you. But it still helps us a lot. It takes a lot of the complexity of building a model away and really allows us to focus on the translation of biology into these individual properties of ion channels without having to worry about how to solve this specifically numerically. And it also provides various ways to visualize the results and just make it easier to, for instance, validate the model. And so an example of MyoKit will be available at the end of the podcast, and there I will show how this can be used to, for instance, simulate the effects of a mutation in the IKR channel. I have a brief slide here that should look somewhat familiar now, and it's also meant to say that it's not that complicated. We have a sodium current, and again, here we have the different doors. Remember the airport, 2 doors opening and closing. These were these M and H processes which reflect activation and inactivation of this specific channel. And these channels are voltage dependent, and that's why the properties of these 2 gates depend on the membrane potential. Now, how this relationship needs to be simulated, MyoKit takes care of that. All we need to do is to specify how the channel responds, for instance, in response to changes in membrane potential in this case. And then we can simulate action potentials as the one shown here. So now we have some terminology. We know that there are ion channels. We know that there are 3 ways to simulate them. We know that we can integrate these ion channels into action potential models, and that there are various tools available to simulate this. Where do we go from here? What are some of the future challenges in this field? Well, many models have been made. And here I've just given you an overview of some of the models that are available for the human atrial cardiomyocyte. And one can see all the different ion currents that are involved. Most of them are red, which means they're present in all. But you can see that there are certain differences between the models. And even when they both have a certain current, maybe the properties of these currents are different. And that results in quite different action potentials that are simulated with these models, although they all reflect the human atrial cardiomyocyte. If you look at the difference in calcium transients, this is even bigger. So which one is correct? They're all based on experimental data. They're all well validated. But of course, all of these models were created for specific purposes. They were made to test specific hypotheses. And therefore, not every model is equally suitable for every research question that you might want to throw at it. For instance, the Grandi model, published in Circulation Research in 2011, was specifically developed to simulate human atrial calcium handling. So if you have questions about that, that is probably the human atrial cardiomyocyte model that you want to start to look into. But this model was not extensively validated for re-entry arrhythmias. And so maybe there's other models that might be more suitable for that. So depending on the question that you want answered, different models might be good for different purposes. And indeed, we now have models for the human atrial myocyte, but there's still a lot of properties that we need to look into. Action potentials are different between different regions of the heart, the atrium and the ventricle. And that is something that we've already included in the models. But what we haven't paid a lot of attention to is that also the subcellular structure between the atria and the ventricle is different, and that this might have an impact on the calcium handling, for instance. Just to show you this, I will give you an example of the structures of some models. Here you again have the Luer and Rudy model that I told you was the basis for a lot of the models. And we have a human atrial cardiomyocyte model published by Courtemanche in 1998. And although you might identify some subtle differences between the pictures linked left to the left and right, you can see that the overall model structure is very similar, despite the fact that we know that there are very important structural differences between ventricular and atrial cardiomyocytes. So we have recently tried to incorporate some of these subcellular structural differences by really slicing up our cardiomyocyte in both longitudinal and transversal direction that allowed us to locally simulate the calcium handling. And with that, we produced a model that was really able to look at spontaneous calcium releases. And so you can see these calcium waves. This would be looking at the top of your virtual cardiomyocyte. And you can see that over time we get these calcium waves propagating through the cardiomyocyte. And we could show that depending on different things that go wrong under disease conditions, in this case, in the case of atrial fibrillation, the properties of these waves change. This only works if you have a model that incorporates the structure of the atrial cardiomyocyte. So again, depending on the questions that you want answered, different models might be better for specific purposes. Of course, this is still very much a simplification. As I said, we like to depict channels like this, or in this case, a renin receptor like this. This schematic is already a little bit more accurate, but it's still a huge simplification. These are big macromolecular complexes. There are a lot of proteins involved. There's different phosphorylation sites involved, and all sorts of ways in which this channel can be regulated. And most of these nuances are currently not incorporated into our computational models. So that's certainly a future challenge. At this point, we also consider all ion channels to act independently from one another. But we actually know that most ion channels gate in clusters, particularly for the renin receptors. Again, this is something that's typically not addressed in computational models. And finally, I already told you about expression and conductance, and I told you that we consider the number of ion channels fixed. Well, if we're interested in a disease process, we know that the expression of many channels changes over time. So how this is regulated is also something that we do not yet have in current models. Nonetheless, I hope that I've given you an overview that computational modeling can be useful. Regulation of cardiac electrophysiology is complex. Computational models can help us a little bit with this. These findings already have some clinically relevant implications, both at the drug discovery level and at the risk stratification levels. I've highlighted that they're basically large systems of differential equations. We follow things over time. They have a very rich history already since the 1950s, and their complexity keeps increasing, but they're very much developed for specific purposes. Nonetheless, I believe that the tools that are now available will help to make them more accessible to a broader audience and also hopefully have some use in your own research. Of course, many challenges remain, but we're working hard to address some of those. With that, I would like to thank all the collaborators that I've been working on in various modeling studies and the Dutch Organization for Scientific Research for funding. And I thank you for your attention. In the next few minutes, I will give you a demonstration of how to use the MyoKit tool to simulate cardiac action potentials. The starting point for this is to go to the MyoKit website, www.myokit.org. And here you can download the software. And the download and install page, there are instructions to either install it on a Mac or on a Windows. For example, if you go to the Windows version, it will tell you to download two components, WinPython, and then the actual MyoKit installer. I have already done this. They're saved in the file in this folder here. Once you've installed both of these components, WinPython and then MyoKit, you can run the program. It should look like this. We now have MyoKit version 1.24 running. And you can see it's still empty right now, but we have 3 main tabs. We have the model definition, where all the individual ion currents will be listed for the model. We have the protocol definition, which tells us how we're going to stimulate this virtual cardiomyocyte. And we now have a default embedded script, which will control how we visualize some of the results. For this example, I want to use a model that's already published and that's available as an example on the MyoKit website. So I will go back to the MyoKit website. I will go to examples here. And we're going to use the model developed by Tom O'Hara and the group of Yoram Rudi of the human ventricular cardiomyocyte. So I will scroll down to the O since they're alphabetically listed. And here we have the ORD 2011 model. It can be downloaded through this link, which I've already done. And again, saved it in the same folder. You can see it's a .mmt file that captures this model. So I can open this file in MyoKit, like this. And now we have a lot of data in these individual tabs, the model definition, protocol definition, and the embedded script. And before we go into details, I just want to show you the default output of this model, which can be obtained either by pressing F5 or by pressing this button that says Run Embedded Script. If we do this, the MyoKit simulation will begin, and it will calculate this beautiful human ventricular cardiomyocyte action potential. Now, how did we get to this, and how does MyoKit do this, and what can we change? Which changes can we make? That's what I want to show you based on this example. We are now in the Embedded Script tab, and I briefly want to run you through the code that is shown here. You can see it's already well documented. Here, the thing's shown in blue. And it tells you that we first are going to get the model from this Model Definition tab, and that we can set which type of model we're actually simulating, whether we want to simulate an endocardial, epicardial, or mid-myocardial myocyte. We then set our pacing protocol, and we give it a cycle length of 1,000 milliseconds. We start the simulation with this model M and this pacing protocol P, and we pre-pace the myocyte for 100 stimulations. And then we actually start the simulation that will be logged, and we store these data in the variable D. Finally, we will plot this, creating a figure and plotting the variable time from the model against the membrane potential. So now how can we use this to make changes? I will show you that with just a few lines of code, we can, for instance, now simulate what happens if we change the rapidly activating delayed rectifier potassium current. I will go after the line where we have obtained the data in the original model. And I will add the lines that will simulate the condition with reduced IKR. First, I have to reset the model. And then I can run it again, storing the data in a different variable, called D2 in this case. And of course, I somehow have to tell the model that I'm changing the IKR. I can do that by saying set constant. And now I have to tell it what I want to change. So how do I know what I want to change? For that, we need to go back to the Model Definition tab. And in Model Components, you can see all the individual currents that are integrated into this model. So I already told you we wanted to change IKR. So I click on IKR, and I get some information about how this specific current was implemented. The main thing that I want to change is the number of channels, or the maximal conductance. And you can see that that is controlled by this variable called base, which has a default value of 0.046. So if I change this variable, this will change the expression of the IKR channel. So this is what I will enter here in my set constant. I will tell it that I want to change from IKR, the base variable. And for instance, I want to change it to 0.02. So that's about half of what the original value was. Well, then all I need to do is actually plot these data. So I'll add in the plot statement the data for D2. Again, I want to show engine.time. and I want to show the membrane potential. And now, if I run the simulation again, it will show us two action potentials. In blue, the control one, and in green, the action potential that you get if the expression of the IKR channel is reduced by about 50%. And I think this is a first example to show you how you can change individual currents and look at the eventual effects on the action potential. And this could be done at different pacing rates, for instance, by changing the BCL variable or by changing other components. Of course, there's also other models available and the tutorials that are available on the MyoKit website. Scroll up here again. You can see that there's also certain tutorials available that will tell you how to make further changes and also how to, for instance, import additional variables from the CellML database. So if the standard example is not yet available, you can get additional models there. So with that, I hope you've had an opportunity to see how it is possible to get more insight into cardiac electrophysiology using the MyoKit tool.
So thank you for giving us the introduction, the demonstration, and thank you for all your efforts.
Thank you for this opportunity.
And thank you for listening to this podcast by the Scientists of Tomorrow.